Hello. Well, thank you all for coming. For those who don't know me, I'm a new hire in the Department of Pediatrics. And today I have the distinct privilege and honor of introducing Michael Clark, who was kind of my former mentor at Stanford. Dr. Clark is the professor of internal medicine, the Carroll and Avis Beekhouse Endowed Professorship in Cancer Biology, and the Associate Director of the Stanford Institute for Stem Cell Biology and Regenerative Medicine. He did his BA and MD from Indiana University way before I was born, but he has had an absolute tremendous effect through the career of many folks sitting here, as well as pretty much around the world. Mike has had a long-standing interest in self-renewal, in particularly the molecular regulation of self-renewal of somatic stem cells, and made the first comprehensive hematopoietic stem cell HSC cDNA library where he identified BMI1, which was the first somatic stem cell self-renewal gene in both hematopoietic stem cells as well, or identified as one of the first stem cell genes in HSCs as well as neural stem cells. He was also the first one to identify cancer stem cells in solid tumors, but more importantly, among all this phenomenal science that he's done, he's always been an incredible mentor to anybody who has gone to him for advice. And that's actually what I will always remember him, his advice that he gave me through my association with him. And one thing I will always remember, he always used to say, never let grants get in the way of good science. So with that, I will introduce Mike. The floor is all yours. Thank you.
Thanks, Sid. It's really great to be here. I love Colorado and the Intermountain West, and I threatened to move here for the to the Intermountain West for the last 30 years. And after spending, Sid and I were both in a meeting in Montana last week, and I fished my way to Denver. And after that, I'm wondering why I never moved anyway. So, and I'd like to thank Rajiv and Sid for inviting me. Now, the talk is going to seem to be pretty disjointed. I'm going to jump between different tissues, but the common thread in this is that, as Sid said, my lab studies the molecular regulation of stem cells, and as I tell folks in the lab, if you find a gene that regulates stem cells, I guarantee you that there's some disease that intersects with that gene. It's often cancer, but it's also very frequently degenerative diseases. And really the common thread is that these molecular regulators of stem cells are shared with many tissues, and that if these stem cell regulatory pathways are perturbed, it can lead to pathologies that we're all interested in, and different pathologies in different tissues. And so with that, the first thing I really want to do is to define stem cells. So probably my most unfavorite term in the world is stem-like, and I just had this conversation. A cell is either a stem cell or not a stem cell, and it's like pregnant. Somebody's pregnant or they're not. There is no such thing as pregnant-like. I don't think, unless you're a psychotic male, I guess. And so I'm going to give my definition of a stem cell, and you guys are lucky enough to have Craig Jordan, who's one of the best stem cell biologists in the world, and I'll let Craig chime in. But basically, stem cells in the blood system, and this is really the prototype. We still know more about blood stem cells than we know of any other type of stem cells in the body. And in the blood system, the blood stem cell has, what really defines it is this property called self-renewal. And what this means is, if you lethally irradiate a mammal, a mouse or a human, the reason, with the minimal lethal dose of irradiation, the reason the mammal dies is because the radiation has killed the stem cells, and the stem cells make the blood, the mature blood cells in the organism, particularly red blood cells and white blood cells, platelets. And those cells are extinguished, and after the lethal dose of irradiation, about 2 weeks later, there's no white cells or platelets to prevent bleeding or to fight diseases, and the organism dies. So if you take that lethally, and the way that this blood system is organized is Rube Goldberg couldn't have made a more complicated system. And it's not that when you lethally irradiate someone, it's not that any cell in the blood system can regenerate the blood system. It's only the hematopoietic stem cell. And so basically, you can lethally irradiate a mouse or a human, take a single hematopoietic stem cell, and because it has the property of self-renewal, that cell, when you transplant it, will find its niche mostly in the adults, in the bone marrow. It will generate more stem cells. Those other stem cells will then leave the niche, go into the blood system, re-enter the bone marrow, find a niche, and start generating more stem cells and also more daughter cells until the blood system is regenerated. And so basically, the definition of a stem cell is a cell that that can self-renew, and my definition of a self-renewing cell division is that the daughter cell has all the differentiation capacity, can make all the cells as the parental cell, and it has much or all of the replication capacity. So you can take a single hematopoietic stem cell, transplant it into a mouse, it'll make trillions upon trillions of progeny, thousands more stem cells. You can take, from that transplanted mouse, you can take another single hematopoietic stem cell and transplant it into another animal, and it will expand the stem cell pool and regenerate the stem cell pool. But it also can differentiate into progeny. And the most immature progeny on the differentiation scale is what we call a multipotent progenitor cell. And these cells also have the capacity to make all the mature cells in the blood system, but they lack the ability to self-renew. And you say, so what? They can make all the progeny. So what happens if you transplant 10,000 multipotent progenitor cells into a lethally irradiated mouse? It will regenerate the blood, those cells will regenerate the blood system. But because they can't self-renew, those cells will eventually extinguish. They lose their replication capacity. So they have a defined number of replications that they can go through, and then they stop. And what will happen is the mouse will survive the radiation, but 2 months later, or 3 months later, or 4 months later, in a defined time period, all these cells will have you'll have lost all the multipotent progenitor cells, and the mouse will then die delayed death. And so the point of all this is self-renewal is a very special type of proliferation. And just because a cell forms a clone in a tissue culture dish doesn't mean it's self-renewed. You have to define, it has to fit this definition. It's regenerated an exact copy of itself. both with proliferation capacity and differentiation capacity. And in the blood, it's the only cell that can do this. So sorry for the long and involved definition, but that definition is necessary for the rest of the talk. Now, back in 2003, as Sid talked about, I figured out how to make cDNA libraries from very small numbers of cells from hematopoietic stem cells, and using some dinosaur technology, we were able to isolate genes that were expressed by hematopoietic stem cells, but not by multipotent progenitor cells. We actually found 21 genes, and the one that piqued our interest the most was a gene called BMI1. And the reason it piqued our interest, I'll show you in a second. And it turns out BMI1 is absolutely required for self-renewal of hematopoietic stem cells. If you knock out BMI1, when the stem cells divide, they make a multipotent progenitor cell. Those multipotent progenitor cells will make mature cells for about 2 months, and then at the age of 2 months, those cells are exhausted and the mouse dies. It turns out BMI1 is also required for self-renewal of several other stem cell types, including the brain, brain, prostate, breast, are some of the, and mesenchymal stem cells all rely on the BMI1 pathway to maintain themselves for the life of the organism. And that's going to be important important for the rest of the talk. And so the reason BMI1 the reason we were so excited about it is it was a member of the polycomb family, and polycomb genes are developmental genes, and they regulate differentiation and cell fate specifications. And they do it by their chromatin modifiers. They're part of the, BMI1 is part of this complex. And what it does is ubiquinates histone H2A on lysine 119, and this shuts down expression of target genes. And the initial target genes that Angie identified is it shut down the P16-4A pathway, and that's involved in cellular senescence. So BMI1 was specifically shutting down senescence pathways in stem cells. It also shuts down expression of P19-ARF in humans, P14-ARF, and P19-ARF regulates apoptosis through the P53 pathway, and we could show that in P53 knockout mice that those mice had partial resistance to apoptosis. Now, this is work of Balaji Kala. Balaji was an MD PhD student in the lab. He's actually immigrated to the U.S. from one of those SO countries, Nigeria, to be precise. And Balaji made what I think is one of the most important findings ever that came out of the lab, despite the fact that he came from Nigeria, or probably because he came from Nigeria. But anyway, what Balaji did is he asked a question: what are these BMI1 target genes? How are they regulating stem cells? And to make a long story short, he found that when you mutated one or two of these BMI1 target genes, it didn't perturb stem cells or stem cell activity in the bone marrow at all. But when he mutated 3 tumor suppressor targets that are being repressed in stem cells whose expression is being expressed in stem cells, he got a 13-fold increase in stem cell activity. And basically what he found is that wild-type multipotent progenitor cells, you just look for black dots here, what he found is wild-type multipotent progenitor cells extinguish and no longer contribute to the blood after 2 or 3 months. And that's, Craig's done, I'm sure Craig's done these experiments. There's hundreds, if not thousands, of labs around the world that have replicated that experiment. But what he found is that the expansion and the apparent expansion in the stem cell pool was not because there was expansion of true hematopoietic stem cells, but with these mutations, multipotent progenitor cells now gain the ability to self-renew. And what this means is that these tumor suppressors are regulating self-renewal. But what he found is if you took one stage cell differentiation down the differentiation pathway, and this is the common myeloid progenitors, that even in the face of mutation of these 3 genes, these genes don't, these cells cannot self-renew. And that implies that the more differentiated a cell becomes, the more genes are involved in preventing that cell from self-renewing. And that has huge implications in cancer. And basically, what Balaji showed is that BMI1 is enabling self-renewal in hematopoietic stem cells by blocking P16-INK4A, by blocking expression of P19ARF, and independently blocking the P53 pathway downstream of P19ARF, and that this function is critical for stem cells to maintain themselves. So, and I think I've gone through all this, and so I'm going to go on to, I'm sorry for the long background, but I wanted to explain both the definition of stem cells, what a stem cell is, and how this molecular regulation works. And what I'm going to talk about now is I'm going to talk a little bit of published work, and much of what I talk about from now on is unpublished in papers that we're just getting ready to submit. So as I told you, BMI1 is part of a complex which ubiquinates his H2A K119, and Madalena Adorno, a really talented postdoc, and if I'm talking about somebody's work, by definition, they must have been a talented postdoc. But so what Madalena came and she wanted to ask, well, if BMI1 is ubiquitinating histones, is there a deubiquinase that it's removing that mark during differentiation, and is this ubiquinase preventing self-renewal of progenitor cells? And so she did a screen of hematopoietic stem cells and multipotent progenitor cells, and she found one deubiquinase, USP16, which I'm not going to go through the data, which actually does deubiquinate the K119 and BMI1 target genes. Expression was a 6-fold higher in hematopoietic stem cells compared to MPPs. Now, we've published that USP16 inhibits self-renewal, but in that paper, for simplicity, we didn't say how we found it, and the way we found it, this is how we found it, and this is also unpublished and will remain unpublished because we're not pursuing it. But basically, what Madalena was able to show is if you express USP16 levels at the same level in hematopoietic stem cells that it's expressed in multipotent progenitor cells, you totally block the ability of these cells to self-renew. And so what this showed was that BMI1 is absolutely required for self-renewal of hematopoietic stem cells. Its levels fall during differentiation. At the same time, negative regulators of the BMI1 pathway, their expression rises in differentiated cells, and that these genes are inhibiting BMI1 and preventing its self-renewal. So what this means is that stem cells can be defined by two properties. First, by expression of genes that are necessary for self-renewal, and lack of expression of genes that drive differentiation, apoptosis, and senescence pathway. And this is important because when you read a paper and you say, well, this cell, this breast cell or this hematopoietic cell, be it normal or cancer, it expresses BMI1, therefore it's stem-like. And again, it's not pregnant-like. It's either a stem cell or it's not. And just because it's expressing a gene that's necessary for stem cells doesn't mean that that cell is truly a stem cell. So to call a cell a stem cell functional, you need functional experiments showing that cell has stem cell properties. And again, so when you read, and this is especially rampant in cancer biology papers, that where the definition that people use for stem cells or cancer stem cells is nowhere near the same as mine, and it's problematic for the field, but I don't know how to fix it. So anyway, now, so what are the implications of this? As I told you, if you find stem cell self-renewal genes, it's pretty certain that you're going to find perturbations of those genes are involved in cancer, and are in some kind of disease, be it cancer or degenerative disease. And what Madalena noticed right away when she found USP16 was inhibiting self-renewal is that USP16 is on chromosome, and this is published, so I'm going to go through it pretty quickly. It's on chromosome 21, and it turns out that it's right next to APP. So genetically, it's difficult, if not impossible, to distinguish USP16 and APP in genetic studies because they're only 3 to 5 megabases apart in the genome, and you have no disequilibrium on amplifications or translocations. And so it's on chromosome 21 right next to APP, and that's going to also be important for this talk. And so we immediately wondered, well, we already know if we increase USP16 levels by a third, we seriously impact self-renewal. And so are there self-renewal defects in Down syndrome in the tissues that BMI1 is important that could also, that could explain at least some of the things that are happening in Down syndrome. And I throw this slide in. This is actually Sid Jaiswal. I'm not Sid. This is Sid Mitra's actually experiment. So in my lab, we found that in Down syndrome, that breast, blood, and other cells that were impacted by BMI1, that there was stem cell defects. And whenever we see something that's important, I always want to see it replicated before the paper goes out. And also, Sid was the best neural stem cell biologist at Stanford. And so we went down to Sid and said, can you look at the neural stem cells in these Down syndrome mice, and can you do assays in human neural stem cells to see if USP16 is impacting stem cell function in both the mouse model and in human neural stem cells. And what Sid found is that indeed there was a neural stem cell defect in this Down syndrome mouse model. He found that defect was largely, not completely, rescued by making the cells 2N or euploid, USP16. USP16, and so basically, even though just by correcting one of the something like 190 genes that are amplified, or that there's a third copy of in this mouse model, that rescued at least part of the pathology, not all of it. This for sure is not the only mechanism of defects in Down syndrome. I'm sure there's multiple ones, but it is one for sure. And we are able to see that, and I'm going to show you more data in human Down syndrome tissue, straight from patients too. I'll show you some unpublished data. And basically, when SID expressed USP16 in normal human neural stem cells, it also inhibited, or it also dampened their ability to self-renew. So, and so what we haven't published, I'm going to go past this, what we haven't published, it also at least somewhat improves the cognitive defect in this mouse model of Down syndrome. And again, the IQ test for a mouse is much different than an IQ test for humans, and I would be shocked if impacting USP16 completely rescued cognitive defects, but in combination with other factors, it certainly could help. But there is no doubt that even though these mice, when they're made euploid or 2N for USP16, even though their cognitive function is improved, that's certain. And so, kind of in summary, what it looks like is that CDKN2A is a critical regulator of senescence and apoptosis in stem cells, and by modulating its expression by these epigenetic regulators, you can impact stem cell function in a major way. Now, we've been interested in what are some of the other mechanisms of doing this. You certainly can't inhibit, you can't wipe out CDKN2A mutation in humans to improve that. That would lead to just a myriad of different cancers. So we wanted to understand what's impacted. And basically, when we looked at USP16 haploinsufficient cells and as I'll show you later, and Down syndrome, both human and mice, we find that there's an impact on the Wnt signaling pathway that's mediated by R-spondin 1 and 2. And USP16 is a major regulator of that, and it's impacting expression of RSPO 1 and 2, and its ligand and their ligand and the LGR5, which is a ligand, which amplifies Wnt signaling. And that's a duplicate. And basically, when we looked at Down syndrome, we also saw a defect in the Wnt signaling pathway in the mice, and just simply making them haploinsufficient, euploid or 2N for USP16, we can largely replicate it. And then we went to humans, and human Down syndrome mesenchymal cells. We obviously can't ethically get neural stem cells from Down syndrome kids and mammary gland stem cells in the other pathway. But since BMI1 and USP16 are working on mesenchymal stem cells, we could study them, and we found there's a defect in Wnt signaling, and this defect is regulated by axin 2. So I didn't show all the molecular data with that study, but basically in Down syndrome, we find for this part we find multiple blood defects in blood, mesenchymal and brain stem cells. It's much of the stem cell defect is the result of trisomy USP16, making them 2N for USP16 rescues that defect, and BMI1 enhances, and USP16 CDKN2A represses the Wnt pathway. I didn't show it the data, but it's actually CDKN2A, which really shocked me, is the regulator of R-spondin. So the target, so it's not USP16 or BMI1 themselves that are regulating R-spondin, it's actually CDKN2A. I didn't show that data. And then increasing Wnt signaling rescues the stem cell proliferation defects, and most, at least some of them, and most importantly, it's R-spondin. And so we actually have the molecule that's doing it. And so, so it's conceivable that R-spondin agonists might improve some defects. Now I'm going to switch gears a little bit. And that's Down syndrome patients have a very high incidence of Alzheimer's disease, and one of the mechanisms is felt to be, and probably is important, is trisomy of APP. But as it turns out that, and there are 30 known APP mutations found in Alzheimer's disease. Now, while it's true that trisomy APP is trisomic in Down syndrome, there are instances of trisomy APP alone, just the small region of chromosome 21 being amplified, so there's an extra allele, so there's a third allele. And those patients have a higher risk of Down syndrome. But as it turns out, as I talked about, that allele, APP and USP16 are essentially adjacent to each other. And when I talked to when I called Dr. Arden, who's done a lot of the Down syndrome and AD genetic and Alzheimer's disease genomic studies, and asked him, when you have trisomy of, when you lose APP and Down syndrome, are there any instances where you've lost APP and not USP16? And he couldn't find any. And more importantly, when you look in the patients in your data sets that have triplication of APP in Alzheimer's disease, do any of them, do they also have triplication of USP16? And he said, in all cases, both genes are amplified. And so the genetic studies, I think, are true, but that left the possibility genetically that USP16 could also be playing a role, and so we decided to look at it. So this is data from the Human Protein Atlas, and I don't know if you all know the Human Protein Atlas, but basically they've generated antibodies against most known human proteins, and protein expression is important, as important as gene expression. And basically, what they found is that BMI1 and USP16 in humans are expressed in mature cortex and mature hippocampal neurons. And so what happens with BMI1 is that the expression is highest in stem cells. During differentiation in the proliferation, in the proliferating cells, the gene turns off, And then when long-lived cells like neurons fully differentiate, they re-express BMI1. And so both of these, so basically BMI1 high expression is in the most immature cells in the stem cell compartment, and they're long-lived, and it also turns back on and is very high in long-lived mature tissues. And that intrigued us because because the pathology in the end and Alzheimer's disease is mature cortical neurons. You lose those. But also and this isn't well known, but the Mayo Clinic, when looking at tests, clinical tests for predicting patients who are going to develop Alzheimer's disease, they found the most highly predictive test for risk factor for developing Alzheimer's disease 10 years later was lack of olfaction or the sense of smell. And it turns out that mature neurons for smell are constantly being regenerated by subventricular zone neural stem cells. And so two pathologies in Alzheimer's disease were in cells that BMI1 USP16 axis. We knew that it was important for those cells functioning normally, and so that really decided for us to look. So one of the hallmarks of Alzheimer's disease is development of inflammation, and what we see is microglia in an Alzheimer's mouse model. You get expansion of activated microglia, and I'll show you in a minute. You also get expansion of inflammatory cytokines, and what we find is that concurrent with that is, and this just measures it. What concurrent with that is increased expression of CDKN2A. And as I told you, CDKN2A is actually encodes two genes in humans, P14ARF and INK4A, in mice, P19ARF and INK4A, which I said is critical for stem cell function and through senescence and apoptosis. So again, we're even more intrigued. So we looked, and this is well known that there's increased apoptosis in cortical neurons in both human patients with Alzheimer's disease, but also in the mouse model. And knowing that this could be working through CDKN2A, we made the Alzheimer mouse mice haploinsufficient for USP16, and we're able to rescue the apoptosis defect. We next turned our attention to the neural stem cell compartment, and we find that there's hyperproliferation of the neural progenitor cells in the Alzheimer's mouse model, and we find that they have a neural stem cell defect. In fact, we find that the neural stem cells in these Alzheimer's models are severely impacted. And what really was interesting is that the neural stem cell defect precedes plaque formation and inflammation. And so in the young mice, when the stem cell defect first starts appearing, this is happening before inflammation has started, at least any inflammation that we're able to measure. And when we mutate CDKN2A in these mice, we can rescue the stem cell defect, but as I said earlier, eliminating CDKN2A function would be lethal. You could guarantee just a plethora of cancers within a year or two. By reducing USP16 levels, we're able to largely also not only rescue the cortical apoptosis defect, but we're also able to rescue the self-renewal defect, at least partially rescue it, just by making them haploinsufficient for USP16. And we can rescue the learning defect, both a NOR defect, which is a measure of cortical and hippocampal function, and also in a Barnes-Maid's test, which is more hippocampal. So we think what's going on, and again, just like what we think is going on is that, at least with mutant APP, you have an early stem cell defect, which is dependent on APP, and it's causing hyperproliferation. I didn't show you this data, but BMI1 levels are falling. I did show CDKN2A. CDKN2A levels are rising. So are USP16 levels. So you're getting stem cell exhaustion, neurocortical apoptosis, and memory impairment. Now, I didn't show this data, but you still get plaques, you still get inflammation. And so you've corrected, at least partially corrected, the apoptosis and the neural stem cell defects, but you still have pathology going on. And so again, I want to emphasize, I don't think by targeting USP16, you're going to cure Alzheimer's disease, but I suspect, at least the ongoing hypothesis, that in conjunction with other therapies that are treating the inflammation and plaque, which also are causing some of the problems, that in conjunction, that this may be a useful target in conjunction with other with the other mechanisms. So now I'm going to switch to cancer. And again, what I want to emphasize is these epigenetic mechanisms are used in stem cells and have implication in cancer. And so what I'm going to present, this is published, it's a New England Journal paper by Piero Dell'Erba from a couple of years ago. And so really one of the questions in stem cell biology, and it's a correct one, so what? There's stem cells in cancer. Does that mean anything? Does that mean anything therapeutically? And what Piero showed is that he could isolate colon cancer stem cells using the markers CD44 and ALCAM. And if you inject the 44 positive, ALCAM positive cells in a mouse, they form tumors, whereas cells on the other side of the mouse, they don't. Now, there's issues with this. This is human cells in a mouse model, not the same environment. But since this work was published with lineage tracing, lineage tracing, the evidence of stem cells in multiple types of cancers, colon cancer, breast cancer, skin cancers, glioblastoma, medulloblastoma, a whole plethora of lineage tracing experiments have demonstrated that there are minor population of stem cells in most cancers. And probably the most definitive one is an agnostic one by Jaco, where he did an image analysis where he could show that in a mouse model of tumors, unperturbed in any way, that he could show that only a minor subset of the cancer cells were clonogenic, and then he could flip the color of those clonogenic tumors. So all the cells were, so he switched cells from being just red in that clone to red, green, yellow in blue, and he could show that the same thing was happening, just a small subset of cells were clonogenic, and most cells had either little, they could form a small colony and then just like a multipotent progenitor cell go on to die, or they never proliferated at all. And so I think there's no doubt that about the model of cancer stem cells. And this is data from Piero Delerba, Tomer Kalinsky, and Steve Quake, where Steve and Tomer did single cell sequencing, and Piero isolated a single cancer cell, labeled it with GFP, a CD44 positive cell, and Tomer and Steve were able to show that this single cell regenerated all the different subpopulations of cancer cells. If you see a different pattern in this, it's a different cell type. And most importantly, it was generating a majority of this single cell that were keratin 20 positive, that in lineage tracing, these cells have little or no ability to reform a tumor, and then a minor subpopulation of immature cells. And so these single cells are regenerating the stem cell and the non-stem cell population. So this cell was pregnant. There's no doubt. So by my definition, it's a stem cell. It's not stem-like, it's pregnant. So, and so using this data, is there a therapeutic implication for this? And this is work from Piero Delerba and Dabashi Sahu, who was also at the meeting in Montana last week. And basically, Dabashi developed a very clever way of deconvoluting gene expression data. And basically, what he found is that the most immature cell in these tumors was ALCAM positive, CDX2 negative. And so these are the most immature cells. I'm not going to go through the algorithm. It's published. It's a New England Journal paper from a couple of years ago. But most importantly, what Dabashi was able to show is that in early stage colon cancer patients, which aren't getting adjuvant therapy because it hasn't been shown to be effective, he found that about 5 to 7% of those patients had tumors that were 100% stem cells. Now, you would predict that that couldn't be good. And in fact, that was the case. Most of those patients went on to relapse unless they got adjuvant chemotherapy. So what this suggests is there's actually a subgroup of colon cancer patients who are not being treated, who should be treated, and based on their stem cell content, and that they may actually benefit from chemotherapy. So I'm going to go fairly quickly here and just show one more epigenetic regulator, again, along the same line. And this is work of mostly Shan Kai. And what Shang showed, in the normal mammary gland, there's a mixture of probably 10 or 12 different cell populations. And basically what he found is that there is a CD66 low cell minus the low cell, and you can substitute EpCAM or E-coherin for this. The markers are expressed concordantly as far as we can tell, and that there's a small subset of these cells that have stem cell activity and regenerate all the other cells in the tumor. So there's a second population of cells that express E-coherin and also have stem cell activity and regenerate all the cells. And these two cell populations regenerate themselves. They're both basal cells. One is more EMT-like, and I'll be honest, I don't know what the definition of EMT is, and I'm going to leave that to the EMT folks. They're both mesenchymal cell populations. One is more and one's less mesenchymal. But not all the more mesenchymal cells have stem cell activity. we see no activity in the epithelial epithelial cells, and some of the more mesenchymal cells also have stem cell activity, but not all these cells have activity. We just published a paper. I'm not showing the data saying in human tumors we get activity in two similar cell populations, and it's the same thing. Some of these mesenchymal cells have stem cell activity. Some are more, they're all mesenchymal. Some are more mesenchymal than the others. But if you look at the more differentiated cells, what's going on? So what are these cells? And so again, this is Xiong's work, and basically using the same single cell gene expression data from Tomer and Steve, we noticed that a rare cell expressed the gene BCL11B, and when we looked at proliferation, none of these BCL11B positive cells, they're actually right here, none of them were proliferating. They were all Ki67 negative. And when we looked at proCAR positive cells, which enriches for a proliferating cell that's more mesenchymal, these were proliferating cells. This data is published. We went through a whole series of experiments to show that these BCL11B cells were quiescent, that these cells were quiescent. We also showed that BCL11B was driving quiescence, and BCL11B, like BMI1, is a chromatin modifier. It's a member of the chromatin remodeling complex, and it's driving quiescence. Now, we knew that in a PYMT cancer model that I'm going to talk about, that the cancer cells are no longer going to need BCL11B to self-renew, because genes that BCL11B are regulating for self-renewal are mutated in the PYMT cancer model. But we thought that BCL11B would still be regulating quiescence, and quiescent cells are chemoresistant. So we looked at the PYMT tumors, and sure enough, these tumors, which are mostly epithelial-like, had a rare population of BCL11B high cells, which I would like to say they're quiescent. We have some evidence that they may be, but it's not definitive at this point, so maybe they're quiescent. But what really shocked us is that when we looked at metastases, all the metastatic cells, and these are single metastatic cells in the lung, were BCL11B positive. When we knocked out BCL11B with CRISPR, the tumors grow. In fact, you would predict now the gene that's regulating quiescence is no longer active, and so the tumors should grow faster, and in fact they do. But they don't develop metastases. And when we looked at primary human PDX models, the metastases appear to be enriched for BCL11B positive cells, and these cells appear to be drug resistant to common chemotherapeutic agents that you would predict they would be metastatic for. And so the experiment that we predicted was true, and the result was completely surprising. I'm sure when we submit the grant on this, my comment to Steve is don't let the grant review modify what you do scientifically. But it looks like this is an extremely important gene. And as I told you, these cells are the quote-unquote less mesenchymal cells. Now, It's going to be more complicated because we now have the regulator for the mesenchymal cell. We've got the molecular regulator identified, and that cell population is also important for metastasis. So it's going to be complicated. It's not going to be some simplistic model where EMT is the whole answer of metastasis, or MET is the whole answer for metastasis. I think it's going to be complicated. So to sum up, just one other piece of work, I think what I hope I've convinced you of that these molecular modulators, that these modulators of chromatin structure really tell you different cell populations that are genomically the same, but are very different cells. And that chromatin really, I think, is the next frontier for, it's not working, frontier for degenerative disease and cancer. It turns out many chromatin modifiers now are being identified as being intimately involved in cancer. And because we think chromatin is so important both for normal stem cell biology and these diseases like I've talked about, Down syndrome, Alzheimer's disease, various cancers, that we really need to have better tools for chromatin analysis. And this is published by Mark Zarniger, and this was a nucleic acid paper where we completely re-engineered chromatin analysis. There's not one single step that's the same from ChIP-seq, because when I looked at the biochemistry, it was the most inefficient biochemical reactions I've ever seen in my life, and it required multiple purifications. So in the published protocol, we were down to one isolation, and that was down, and that was after the initial reactions. We've now eliminated the one column purification from this revised protocol. And from the published protocol, we were down to 200 cells that we could do what we call T-cell seq. If we have good antibodies that are labeled correctly, we're now down to 25 cells. And the hope is by just modifying the there's one more chemistry reaction that's inefficient, and it's labeling the antibodies. And we think we know how to fix that. And the goal is to get to a single cell. But just by being able to get to 25 to 100 cells, we can at least do some chromatin analysis on subpopulations that just weren't possible before. And so with that, I think I acknowledged all the people that were involved in the work. And again, anyway, I just really appreciated being here. I'm sorry, I think I went a little long, and I apologize, but if there are any questions. Craig.
Mike, there's as you showed it many years ago, there's this fascinating relationship between the reactive oxygen species low phenotype and stemness. Do you find that metabolic or physiological state is related to regulation of BMI?
You know, there are some indications it is. It turns out that that BMI1, something I didn't show is that mitochondria are intimately involved to unfolded protein response, which also clearly plays a role in Down syndrome, for example, and probably in other diseases, and some other injury responses. That ROS is the unfolded protein response, and some mitochondrial pathways are activated, which activate ROS, and BMI1 and SP16 is part of that feedback loop. So the answer is yes. What we also showed in that paper, in which most people probably don't remember, is that is that we had an ROS high subset of cells with stem cell activity. So it goes along, it goes along with this notion that this idea that stem cells are all the same is ridiculously simple. Stem cells are responding to the environment just like every other cell. So in the blood, it's responding to infections, to bleeding, to inflammation. In the mammary gland, it's responding to estrogen cycle, pregnancy, and evolution. People don't realize this, but 2 of my close relatives had mastitis after pregnancy, and that's going to, and so you're getting huge inflammatory responses through the innate immune response. And we had a paper a while back showing that stem cells are actually, when you get inflammation and innate immunity, you get proliferation. So I think that paper is actually going to end up being tied to the Alzheimer's and Down syndrome stuff. We see hyperproliferation in Down syndrome, and which again is if the unfolded protein, for example, and any inflammation you're getting is going to cause stem cell proliferation, hyperproliferation. Yes.
So there is obviously, I'm sure you're familiar with the Camargo paper showing that MPPs can actually be major contributors to metabolism for quite a while. And there's another paper that lineage tracing that I think showed for at least a year they could be contributing. And I don't know if that would fall into the definition of stem-like, because they may not be stem cells in terms of like for a 2-year lifespan of mouse. But
So again, what that says is those cells can maintain themselves for a period of time. Now, what people are seeing is that with aging, there's mutational events that are going on in stem cells, which is normal stem cells, which is changing their biology, so they can become, for example, myeloid skewed, and which will lead to readouts of the cells predominantly giving rise to myeloid cells. And so part of this is when you do these type tests, because with aging, you're clearly getting mutations which are altering stem cell function. And then Robbie Leggetti, who I guess was just here, Robbie and Steve Quaid showed that with that you can get clonal hematopoiesis that occurs with some mutations, and it turns out most of these mutations are in chromatin modifying genes. And in epigenetic regulators that you can get clonal expansion in. So you have to be very careful. So if a cell is mutated, as I showed you, I can make an MPP into a stem cell. So if you get certain mutations, you can turn a cell into a stem cell. And if the cell is unipotent and it self-renews and it can maintain its differentiation capacity, it's still a stem cell. It's just not the normal young hematopoietic stem cell. And so, and again, if it can replicate for a year, or Weissman used to call these short-term stem cells, there's different names for those, but they clearly exhaust, whereas normal stem cells may or may not exhaust.
Back to the mammary gland and the QIMT model, these BCL11B cells that are rare in the primary tumor and the metastases, all of them are positive?
It's cool, and again, I'll tell you what happens. So initially, they're all BCL11B positive, and they're basal cells. That's at the single 1 or 2 cell level.
Okay, but then they start to form daughter cells?
Yeah, when they form daughter cells, you get the same result as in Piero's experiment with the single colon cancer stem cell. All of a sudden, they generate a tumor that's got all the heterogeneous cell population.
Okay, that's what I was wondering. And those cells, are they better at surviving in the blood do?
You know, I don't know. We're early into this, and as I told you, it's complicated, and I don't know how we're going to publish this in the end. I don't want I don't want to publish a paper that's the results are misleading, but it's going to be confusing to people to say you've got two genes expressed by two polar opposite cells that are absolutely required for metastasis. So the mechanism, the relationship, I can't tell you. All I can tell you is both these genes expressed by clearly two different cell populations are absolutely required, and both these cell populations have stem cell activity, and they regenerate each other.
Those cells that metastasize, they are less epithelial in the sense that they lose estrogen and progesterone receptors, they lose signature. We know that when they land at the metastatic site in the lung, they are ER negative.
Yeah, and so that's the B cell. Both these cell populations are ER/PR negative. They're both basal cells. And again, it becomes a semantic argument. They have basal cell phenotypes, so they're not a true mesenchyme fibroblast, but they're expressing genes that are associated with mesenchyme fibroblasts, but not all the genes. So one of the cells expresses more than the true mesenchyme-derived fibroblast genes than the other. But again, we know there must be an even more differentiated, truly like very mesenchyme-like cell that's lost all proliferation capacity. We've seen that. So it's more nuanced. It's not the simple EMT. It's all EMT cells are stem cells.
So along those lines, you talked about this a little bit when we met. What you're arguing is not completely EMT, it's somewhere along the continuum, right? Where it has some xenomal characteristics, but not others. Is that getting that right?
Well, it's semantics. They're basal cells, and they express, and all basal cells express some mesenchymal genes.
Okay.
So Jean-Paul Thierry is actually has become a very close friend. And when I talk, and I leave the EMT to John, or to JP, and he's really the father of EMT and mammary gland. And basically, I tend to be someone who's very functionally related. These are the genes that are doing that. And if you want to say they're EMT genes, that's not my field.
I guess one of the questions I have is maybe not, but do you think in the case of the tumor, the xenomal tumor organs, or if you're dealing with a sarcoma, that in fact they be moving in the other direction, so they be moving some of their mesenchymal characteristics to be almost xenomal? Does anyone look at that?
I haven't. I don't have the answer to that. So when we first found breast cancer stem cells, some people who are working with sarcomas were friends of mine at Michigan and came down and asked us to see if we could find markers for stem cells in sarcomas, and we couldn't do it. We tried, I don't know, for 6 months or so and couldn't find any markers that were informative. And so I thought, well, maybe this is a tumor where there's just not markers. I was just at a talk a couple of months ago where somebody was doing lineage tracing, and then they could use they could use markers to isolate cells. But these are driven by these are promoter-driven fluorescent markers where they could do it in mouse models. And they found when I looked at the data, which by all criteria looked like a sarcoma stem cell for the type of sarcoma they were looking at, was a single type of sarcoma. And whether all sarcomas fit that model, I don't well, but at least that one appears to, and it was one that we tried to find cell surface markers that we just couldn't. So, unfortunately, cell surface, what I'm seeing is cell surface markers. They're not uniform population cells. So if you take the original markers we used, 44 and 24, and you really look carefully, I mean, that's a mixture of 5 or 6 different cell types, some of which probably have 7 different, some don't. So using that mark, so when I see the argument at 44 or 24, therefore it's a stem cell, I go bananas, because if you read our paper, we said enriched for